of somatic point mutations in mRNAs with amplified single molecular FISH. Dr. Tyagi, please go ahead.
All right. Aniruddha, thank you very much. I really appreciate the opportunity to give this seminar to folks at NCI. And I already see somebody who's really dear to me, Gregoire Bonnet, that I had pleasure to work with many years ago. He just joined in, and I'm so happy to see his mask cover face. So with that, I'll begin. I'm going to talk about our current work on detection of point mutations in cancer. So as perhaps you would be familiar with, number of targeted therapies are now commonplace.
Sarah, you want to just take a look for a sec?
I guess everybody should mute themselves, please. So these targeted therapies for cancer very often require you to analyze the tumor beforehand, because the point of certain mutations, such as in EGFR gene, allow you to adopt certain targeted therapies. And an appearance of other mutations may render them useless. So a analysis of point mutations in the primary tumor is of great importance. And so we have developed a system that would allow you to do it in the first biopsy. And the system is based on in situ fluorescence hybridization. And so this is an overview of my seminar. Hopefully, I should be able to finish it in 45 minutes or so, and we can have questions and answers after that. So I will describe our approach of single molecule FISH, then a recent elaboration of that called amplified single molecule FISH, and I will describe how we use amplified single molecule FISH, or ampFISH, to detect SNPs in messenger RNAs. And then briefly, I will describe our recent effort to do these detections directly from tissue sections obtained from a cancer. So let me begin with the inventor of hybridization. And hybridization is an old technique, first described by Joseph Gall in 1968. And this is him as a young boy in Maryland, I think. I recommend those who are interested in the history of in situ hybridization to look at this biographical article in Nature Medicine on Joseph Gall. So he was the first person to detect nucleic acids, DNA, in frog oocytes. And this picture you see here is of frog oocytes labeled with radioactive ribosomal RNA, which he was able to do. He sort of incubated the oocytes with radioactive ribonucleotides, purified the RNA, labeled RNA now, and hybridized them to fresh oocytes. And these spots you see are amplified genomic loci for ribosomal RNA genes. And this is the basically he hybridized them and took a autoradiogram. And this is the first time people were able to see unique genes in the cell. And with this became a long, glorious history of in situ hybridization. This technique has been in use ever since, and many different innovations have been introduced in this. So radioactive RNA probes were replaced with sort of chromogenic amplification systems where you would attach a enzyme like horseradish peroxidase to a probe, which would give rise to an amplified signal, usually in a bright color. There was tyramide amplification, but these amplification systems have one particular weakness. The amplified signal goes away from the site of your probe. So you get less than accurate detection of where your target was. So you get diffusion of your signals. And next big improvement in this was made by Robert Singer in Albert Einstein College of Medicine. And what he developed was, which I refer to as Singer probes. He essentially would use 50-mer synthetic pieces of DNA, label them with 5 fluorescent dyes, and all of these probes would bind to the same RNA and deposit so much fluorescence that each RNA could be seen as a fine fluorescent spot. And this work goes back to a science paper in 1998. And our contribution to this long march of history of in situ hybridization was to simply modify Robert Singer's probes so that it would be easy to make them. So we basically decided we are going to use oligos, little oligos, 20 mers, label them with just one dye at the 3' end or 5' end. And this allows you to make pools of oligos and label them en masse and purify them by HPLC together. And so basically, this is our system, and it became very popular. And we licensed this technology to a company called BioSearch, and they market these probes as Stellaris probes. Many labs use them, and the advantage is sort of clear from the typical results you get. So here is a bunch of HeLa cells probed for their, we made probes for CFOS gene, just as an example. And so there are 50 oligos that will bind to CFOS RNA. And the way you do these hybridizations is you take your cells, culture them on a cover slip, fix them with formaldehyde, and remove the membrane by incubation with alcohol or detergent. And then you add these probes, wait overnight, and next day you wash off the excess of the probes and you look. And you see these fine spots. Each spot comes from a single molecule. And if you notice, in the nucleus, there are these brighter spots, and these are gene loci where the nascent RNAs are still attached to the gene. And there are multiple copies of them, so you see they're brighter. So you can localize these spots, you can count them, and we estimate about 85 to 90% of all the RNA molecules present in the cell can be detected. So basically, when you formaldehyde fix the cells, a cross-linked matrix of protein forms that traps the RNA. And the probes can go in and out by find the RNA, but RNA itself stays immobilized. So here is this field of cells detecting CFOS RNA, and in the same cell, you can label this sister protein RNA, CJUN, with a different color probes, and you can computationally identify these spots and count them. And this is a typical workflow in our lab and in hundreds of other labs that have adopted this technique. I will just quickly describe to you evidence that we are able to detect single molecules of RNA, and these spots represent those single molecules. So here is a experiment with MAP2 mRNA in the neurons from a rat embryo, and then you can see single spots that are discrete and separate from each other. You could count them, and you could also measure the number of molecules of mRNA by real-time PCR, and the number of mRNA present in a typical cell is comparable to the number of spots you find in those cells. This is another experiment where we induced expression of a gene called FKBP5 and image the mRNA before and after, and you can sort of see the probes ignored all other RNAs in the cell and detected only the induced gene, and as indicated by this change in number of spots. This is yet another experiment. It's shown it's in yeast, and yeast makes this RNA called STL1 only when you add salt. Otherwise, the gene is silent, and you can see the spots only when you add the salt. All this to show that you are detecting your correct RNA. And this is an experiment done by Alexander von Oudenarden in MIT. And they counted the spots for these 5 genes, and also measured measured them by deep sequencing. And then you can see a nice correlation between the two measurements. And so the technique, basically the number of spots is correspond to the number of reads. So here is few additional evidence that what you're detecting is real. If you make probes for something like GFP RNA, it would be expressed only in the cells that get transfected with that, and so on. I will not belabor the point because many labs use it successfully. Again, I'm just recapping the data that I just showed you about the number of molecules number of spots being proportional to the number of molecules found by real-time PCR. So we and many other labs have used this technique very successfully to do all sorts of cool things. And I'll just give you one example where we were able to image splicing in situ and look at both pre-RNA as well as spliced products of the pre-RNAs in the cells. And this is, so basically you can make about 50 oligos for an intron and label them with a green color, 50 oligos for an exon or a group of them, label them with a red color, and the introns will be visible in both colors and the exons only in the red. And you can essentially, even if there are very few spots, few molecules in the cell, you will be able to locate them. This is another effort in the lab to broaden the scope of this technique to flow cytometry, which is very commonly used. And in this case, we probed interferon gamma RNA in PBMCs, cells in the blood, and so typically interferon gamma is not expressed in resting cells, but when you stimulate them with PMA and inomycin mixture, about 15% of the cells begin to express interferon gamma, and you can sort of see 2 cells in this group were making the RNA, and you can, by flow cytometry, you can identify that proportion. However, as I would like to come now to key limitations of this technique, even though you use 50 probes to increase the signal, you um, um, are still limited by the signal intensity. The signals are rather faint. You need high numerical aperture objectives, 60 to 100x magnification, and if the gene is small, many genes are very small, and those would not accommodate 50 oligos. So at least 30 probes are needed for you to detect the spots. And most importantly, from the point of view of this seminar, you can't detect point mutations because your signal comes from binding of about 50 oligos. And so we would like to be able to detect point mutations somehow in the same format. And this is what I'm going to talk about. And so in order to address these needs, many labs have worked out sort of new ways to amplify signal. And one of the ways is so-called branch DNA. So basically you create a little Christmas tree on the top of your probe, and that allows you to have large signal. And there is yet another technique called hybridization chain reaction, and I'm going to describe to you the technique because I'm going to use it in the technology that I will describe now. So basically, let's begin with a little bit about how hybridization chain reaction works. This is cool idea that comes from peers and colleagues in Caltech. And what they did was they assembled these pair of hairpins that are metastable thermodynamically. And these two hairpins, they're sort of mutually complementary for most of their sequence, and yet they don't bind to each other. And that happens because they form very strong hairpins, intramolecular hairpins, and they would stay in a mixture stably until you add a initiator sequence. So if you look at the organization of this hairpins, you would see that this sequence A prime can bind to that A and create a possibility for branch migration in this molecule so that B prime can displace the B prime of the hairpin and sort of open up that hairpin. So once the H1 has the sequence opened, it can interact with the second hairpin in the same way and open that. If once that opens, H1 can interact again, and this goes back and forth until you have creation of a long double-stranded DNA. And since the hairpins are labeled at the end, you actually have many, many fluorescent dyes on this multimeric product. And this multimeric product stays attached to the site where this initiator sequence was. So these folks, Niels Pearce and colleagues, they tried to use this system to detect RNA directly, but there were limitations as to what kind of sequences will be good initiators. You can imagine that a good pair of hairpins should be stable on their own, but will react very rapidly when an initiator is added. And they worked out a system where few hairpin pairs had these characteristics, and at least 5, 10, if not more. And so they decided to use this as a tag on primary probes. So here is your RNA, and here you will bind a RNA specific probe, which has a tag, a 3' tag, which is the HCR initiator. And they found that best way to do this is sort of by tiling these probes, same way as we do it in a single molecule fish, a single oligo is not quite sufficient to give you detection of almost all the RNAs. So basically, you tile your probes, and the hybridization chain reaction will be initiated at each of these sites. So we decided to use this system to detect a little stretch of target sequence in messenger RNAs. And basically, I want to come back, describe to you one of the problems in the system. So this idea of tagging a RNA-specific probe with a tail, even though it would amplify your signal, it creates a problem of high background, because imagine that these probes are bound to a wrong non-specific site. So a non-specific binding event will also create amplified signal. And so as you increase the number of probes in the hybridization, you also increase the probability of creating false signal. So we decided we are going to have a system where the initiator, HCR initiator, is hidden, is sequestered in a hairpin, and it will become available only in response to the target. And the way we engineer this is that you actually have two probes. And so one acceptor probe, and this is where your initiator sequence is, and a donor probe, which is going to bind next to the acceptor probes. And when they bind next to each other, a situation is created where this interaction between these two sequences occurs, which leads to displacement of that green sequence and opening of the initiator sequence. Now you have a target-mediated unmasking of this initiator sequence. So if this probe was bound to a non-specific site, it would be unlikely to also bind to that donor probe, and therefore you will not generate a false signal. And so basically, this is how you'd use. You will wash the excess of these probes, you add HCR hairpins, you develop a signal, and you will detect. And this is a little simple experiment to show you that it actually works and is pretty specific. So here is a bunch of cells, in fact, 3 HeLa cells transfected with a plasmid encoding GFP. And then we hybridized these pair of probes. We washed the excess of the probes, added the 2 hairpins, washed the excess of the hairpins, and looked on the microscope. And you can see the GFP positive cell, meaning this is the one that's expressing the GFP RNA. This is where you can see the spots by HCR, but these other neighbors that didn't get the GFP didn't see. So this is quite specific. In fact, even if you increase the contrast a lot, you won't see any signals in those neighboring cells. However, how do you go about detecting a point mutation? So I would like to point out that these target binding regions are pretty small. So they will be destabilized by the point mutation. So to sort of have a sort of very, very specific detection of the point mutation, we decided we're going to have the point mutation in the donor region of the target, okay? And the way also we wanted to have a green signal for wild type and a red signal for the mutant. So in the same cell, it's likely that both kind of RNAs will be present. And we want to be able to detect both and do it by the color of the probe. So this is sort of a little more elaborate. The probes there are now 4 probes, 2 acceptors and 2 donors, and donors bind to the same site. Acceptors bind next to each other. So this donor site, donor probes will have a choice. Only one of them can bind. So if the left donor binds, it can interact with the left acceptor and give rise to green color using HCR hairpins H1 and H2. And if there was a mutant present, if there was a wild type present, the right donor binds and it interacts with the right probe, and that gives you red signal. So both signals cannot be created by the same RNA. And also, Gregva will appreciate that if you create hairpin-shaped probes, you actually achieve higher specificity than linear probes. So these donor probes are shaped like a hairpin. So basically, they don't bind, they don't spend energy in opening their stem unless the target is perfect. So there are a number of thermodynamic reasons why this thing is pretty good. So to test the system, we had a pair of plasmids that code for GFP, essentially the same sequence, except we created a silent mutation in the GFP, and that became our target. And you can sort of see a mutant RNA shows green signal, green spots, occasional red ones. And similarly, the wild type GFP gives red signals. And the specificity is almost complete. You barely see signals from the wrong target. So this is a pretty good demonstration, except this is completely artificial system. And specifically, you make very large number of RNAs when you transfect cells with the plasmid. The number of copies of a typical messenger RNA in a common system is very low. Like a natural RNA is expressed from 50 to a few hundred copies per cell. And question is this sufficiently sensitive for you to detect detect your natural RNAs? So we adopted the system to detect EGFR messenger RNA, and EGFR is a commonly used target in the clinical diagnostic field for cancer mutations. And so we sort of targeted a very famous mutation, L858R in EGFR. And so the way this experiment is done is slightly involved, and hopefully I will not confuse you. So basically, EGFR is a long RNA. We would detect the body of the RNA using smFISH. So 50 oligos, you can see this guy is labeled in Texas red, white spheres, and these 50 oligos bind to the RNA away from the region which we are going to interrogate with our mutation detection probes. And they will give you a signal in Texas red. And then we're going to detect the mutation L858R using these 4 probes in the same way as we did in the GFP experiment. So when you have, you can either get a green signal or a red signal, Cy5 signal, and you can see all 3 colors in the same cells. So we imaged 3 cell lines. And the top cell line is H1975, and this expresses this mutation at L858R mutation. So let's look at that. And these other 2 cell lines are wild type. They only express the wild type target. And so let's look at Texas red. So this channel will show all the RNAs of EGFR. there are about 50 copies of EGFR in these cells, and you see those spots labeled in Texas red. And now if you look over Cy3 channel, you see only few spots, and this detects the wild type messenger RNA, and Cy5 detects the mutant RNA, and our computational algorithm can find where the spots are and what channels are they positive in, and so you can sort of see this cell line gives many spots that are labeled in Cy5 and Texas red, that would be this sort of gold color, and these other cell lines only show Cy3 signal, and the quantification of this data is presented in these histograms, and you can sort of see these cell line 1975 gives mostly about 70 to 80% mutant, and the rest wild type, and this HeLa and A431 cell line gives mostly wild type. To the extent the purple color is seen here, that's the leakiness or the false signals that you get. However, one thing that is important is that in this cell line 1975, there are 4 copies of EGFR gene, and 3 of them are mutant and 1 of them is wild type. And the messenger RNAs that we detect reflect that ratio. And so this is pretty remarkably high specificity for something that is expressed in 50 copies per cell or less. And so all this data is described in this paper, Maras et al. In PNAS. And so since then, we have been trying to use the system to I would like to come back. Before I go to tumors, I'd like to point out one more thing we can do with the system. So there are several aims we have in front of us. One is to be able to detect point mutations using one pair of primers that bind to the region of the mutation. And the second is to also increase the signal without increasing the background so that we can do flow cytometry for a number of targets. As you all know, flow cytometry is very important and popular technique. And if you could detect detect RNA directly in flow cytometry in a quantitative manner, it allows you to do analysis that have not been feasible before. And this experiment shows the improvement this ampFISH will bring to flow cytometry. So we're detecting, like before, interferon gamma RNA in PBMCs stimulated with ionomycin PMA. And so when you use 50 oligos, simple smFISH oligos, you get a pretty strong signal. And this signal detects the population that are expressing interferon gamma. But you notice that it's barely the population of interferon gamma expressing cells is barely separated from the cells that don't express it. And when you use what I call as passively tagged probes, where you simply attach this tag of HCR initiator to about 48 oligos, you get a huge increase in signal, but you also get a huge increase in background. So, and that's the problem with the simple use of HCR as a tagged as tagged probes, and the inventors of this technique, HCR technique, have recognized that as a problem, and they have a different solution than what we present. And their solution is to actually break the RNA binding region into two sequences, and then create a joint two-part initiator. And this is called HCR version 3. And that is pretty effective, but I would like you to look at the technique we developed, where the two probes come next to each other, unsequester the hairpin, and do it for all 24 pairs on the same RNA. And this gives a improved signal. The signal is not hugely stronger as we had hoped. When we begin to work on this, we had hoped that we will get 20 to 100-fold increase in signal. You don't get that. You get about, in this case, about 5-fold increase in signal, but the background doesn't increase at all. There's one more experiment I think I have here. This is another experiment, same kind of idea that we express GFP transfection of HeLa cells with GFP and compare the signals that you get with smFISH and when you use 24 smFISH probe, you get this kind of signal. So here, this is a unique system because you can detect the transfected cells both by GFP as well as by RNA probes. And the ratio of Cy5, which is RNA, divided by GFP tells you the sensitivity of the detection. So when you use 24 probes, the ratio is 0.4. And when you use 48 probes, the ratio becomes 1, meaning increasing the number of probes makes you more sensitive, indicating that our measurement is able to measure the increased sensitivity. And when you do this with 24 ampFISH pairs, you get about 10-fold increase. So, so in this system, we are able to get about 10-fold increase in signal. So to summarize, we can get about 5 to 10-fold increase in signal. And for a single site where a pair binds, we can get as much signal as 50 smFISH probes. With this, I'm going to present my final set of results. And this relates to actually detecting EGFR mutation L858R in a cancer specimen. This specimen we got from Sridhar Ganeshan and Gregwa, not Gregwa, another guy that whose name would appear in my acknowledgements. Basically, this is a tumor that originated adenocarcinoma, originated in lung, then metastasized to brain, and we obtained sections of this and these little pieces from the tumor, and I had no clue which of these cells are have tumor origin and which are stromal cells. And so first thing is we looked at H&E staining of this section, and I'm going to show you one a piece of this from that region E, and these I learned later are the tumor cells, and I suppose these are not. And so so we imaged this with the before using our probes. Fortunately, there is an antibody that is available for quite a few years that specifically detects EGFR L858 mutation. So it was raised specifically for that mutation. So it will not detect the wild type version of EGFR. So the signal that you see is immunofluorescence from the region E, and you see these sort of signals that are consistent with the tumor region, and they are peripheral. They are on the um, perhaps membrane of the cells. And so this tells you where to expect the signal from ampFISH. And so we did the experiment with the ampFISH. So the green color will detect the wild type, and the red will detect the mutant version. And you can see, so this is adjacent section, not the same section where we did the immunofluorescence. So we are just doing, in this case, ampFISH, no immunofluorescence. It's not multiplexing, and just to make sure that colors don't confuse. And so if you, the green is sort of very faint, most of the signal is the mutant, and it is present in the cytoplasm. This is sort of a good look at that. And so we found the signal in this archived tumor section, and it is present only in the tumor cells. The cells from the stroma don't give this signal, and this is a sort of little scan. But I should say that signals are still very faint. This is not it's not a technique that a high school trained technician can use in a diagnostic lab. But what I would like to say is that the signals are very specific. They're detecting point mutations in the tumor, in the primary tumor, and one more thing to show, I actually took that highly probed section and scratched the material with a blade and extracted the DNA and did digital PCR on two mutations. One L858R, so the top lanes show that, and one, its companion mutation T790M. And this tumor was characterized earlier by deep sequencing to contain both. And so we were able to find that about 75% of the DNA molecules in these regions were mutant for L858R, and a minority was mutant for the T790M. So we have not probed that yet. The work is in progress. So, but we can see that we are able to detect by both technique, droplet digital PCR, as well as by ampFISH, the signals in these sections. And I think this is my last slide. I will summarize. So basically what I'm presenting you is this technique that marries smFISH with HCR. It confers upon the probes the ability, this special feature that the HCR initiator is sequestered and hidden until the probes bind to the target. It makes them really specific, and it allows perhaps uniquely, I don't think there is any other system that allows you to provide an amplified signal, single molecule signal for a point mutation in RNA or even in DNA. And by tiling these pairs of ampFISH, you can increase the signal perhaps by tenfold. And when you do that, you probably will be able to overcome this really pestering problem of autofluorescence in sections. Also increase signal by tiling probes or probe pairs allow you to do 20x imaging. And the images that I just showed you were done at 20x imaging. So the challenge is when you analyze sections, tissue sections, 100x imaging that we have traditionally done is not useful because then you see only one or two cells or few cells. You don't get a sense of the geography of the layout of the section, which is what you need for clinical diagnostics. So you have to be able to do something like 20x imaging for you to get the sense of the geography of the section. And so the results that I showed you with the adenocarcinoma were actually with 20x imaging. And I am also showing that you can do flow cytometry with this technique and perhaps detect a wide variety of RNAs. Perhaps, as long as the expression level is something like 50 copies or more, you should be able to detect them. And with this, I'm going to acknowledge all the people who have done the work. So single molecule FISH or our version of it was invented by Arjun Raj. Many of you know him. He's a star in his field now, and he did his PhD in our lab, and he was a mathematician when he came, but now he's a experimentalist and an expert at that. He's at UPenn. Diana Vargas, she did all the work that led to the smFISH. She created these genes with tandem repeats that we could detect the RNAs with molecular beacons in cells, and that led to the cell paper that I described with the splicing. Salvatore Maras is my long-time colleague. He's expert in all kinds of chemistries with DNA, and on the left is molecular beacons. And then these are my collaborators, Sridhar Ganeshan and Gregory Riedlinger, who gave us these tissue sections, so I think at this point we are funded from a very nice grant from Cancer Institute, and all the work was done at Public Health Research Institute at Rutgers University, and I'm happy to answer the questions that you might have.
Folks, please go ahead and ask questions and unmute yourself. Thank you.
Hi, Sanjay. Great talk as usual.
Oh, Gregor, nice to see you.
Nice to see you. Sorry, we mask, right? But I wanted to ask about the technique to distinguish the two alleles. And you show in this H1975 for EGFR, you had 4 copies, and you could explain, you had 3 well-detected, 3 mutants well-detected and 1 wild-type. Did you see any case of allelic exclusions when you have one gene and mutated genes, and it will be bimodal when you look at the individual cells and which locus is being expressed? You could have argued for that for cytokines, right?
This is a very important question. In this case, I didn't see an obvious pattern. I mean, I didn't quite analyze it for that. Are the individual cells that express only one or the other? Part of the issue is perhaps it didn't stare at me because there's gene amplification. So the probability of exclusion goes down, right? It's when there are multiple copies. But however, recently we have done something interesting similar to that with another gene called COX-2. And this gene, unfortunately, is also present in 4 copies. But Greg, we have a paper in PNAS just published a couple of months ago. I'll send you a link. And there we looked at, by the same technique, transcription side, the cluster of genes cluster of RNAs that are present in the transcription site. And we could distinguish using an SNP two kinds of transcription sites. And there we were able to show that they're binomially distributed, meaning that there's no distinction, there's no exclusion, they're just randomly, they come on and off, at least in that gene. And in fact, we analyzed data in great detail for those 4 copies of COX-2 gene.
I'll check it out. Thanks.
More questions from anybody, please? Go ahead, unmute yourself. Well, while they're thinking, I may ask a pretty general question. This technology is used pretty much everywhere. In clinical setting, how feasible it is to use this technology to diagnose different kinds of, mutations in specific types of cancers.
Yeah, so first of all, I mean, limitations are you must know what mutation you're looking for.
Yeah.
And so for targeted therapies, these things sort of are clear, meaning if you're going to use EGFR drugs, you know what mutations to look for. And so that is not such a serious problem for targeted therapies because the mutations are known. So far, I don't have a clear sense how reliable or feasible it would be, but because I've just done a few sections and not, I knew what to expect there. So ultimately, blinded studies will need to be done. But in terms of imaging, it's not very sophisticated in terms of, like, it doesn't require like specialized microscopes or anything. So it should be feasible. But what I would like to say is that the effort, that you make on this is worthwhile because the primary biopsies that you make, you want to know from that by directly whether the mutations are there, whether the resistant mutation has appeared, and so on. So I think it's a worthy goal to explore this and the utility of this in clinical settings.
Have you contacted any molecular pathologist in your institute or elsewhere?
Yeah, in fact, that has been a big problem. So this technique we have published this several years, 2-3 years ago now, and we contacted all kinds of people, and it was difficult for us to obtain few sections that these folks from CINJ New Brunswick gave us. So I would like, since I'm talking to the right audience, if anybody has access to a possibility of providing us or connecting us to labs that have sections, would be absolutely appreciated, and we would work in a collaborative basis. This really has great potential, and if anybody in the audience can help, I would really appreciate. We need samples and specimens.
Well, I'll keep in mind, and if I find something of relevance to you, I will forward it to you.
Yeah, and also one thing to say is that technique is general. It's not just EGFR or this mutation. We should be able to develop this for essentially any RNA, as long as it's expressed at some reasonable level and not very sparsely expressed. And so, whatever is the favorite system of target, we can work with that.
How many colors have you been able to multiplex? I mean, how many mRNA could you track in the same cells?
Greg, were about 4 RNAs. So the tissue sections have a problem of autofluorescence, especially lung sections, and that throws away half of the spectrum. But in tissue, in cultured cells, we can do 4 colors pretty comfortably.
Yeah, because people have made construct with GFP, RFP, etc. to do like chromatin epigenetic regulation at a given for a given gene and comparing one chromosome versus the other. But like if you could multiplex it, you could really build a model of what's intrinsic for a given gene, what's extrinsic, and redo the whole stochastic gene expression at the level of epigenetics, being able to go beyond.
Yeah. So we should be able to fit immunofluorescence markers for sure, at least 1 or 2. So I think 3, 4 colors are absolutely feasible. And then, in fact, some of the images I showed you had 3 colors, to build controls.
Right.
All right, folks, if you have any more questions, go ahead because we are pretty much down to the end of time. If not, I just want to thank Dr. Tyagi for fantastic, technically sophisticated, as well as really helpful presentation for the researchers as well as for the clinicians. We appreciate it very much. And all of you, those who joined today, thank you very much for joining this presentation. Have a wonderful day.